we're asking about chapter 9, what can we expect? And as I kind of think about chapter 9, you'll probably get one question. Okay. Now, if you read my mind, I'm a structure function kind of guy, right? Right? We learned all the structures of everything so that we could see how does that lead to the structure. And the major single carbohydrate that you have to know the structure for is glucose. So
You need to know the offsets of it, the subsets, the different.
you need to know glucose. Okay, so I'll just narrow it down to for you for this semester. You need to know glucose. How's that sound? Next semester, you have to know all those ones that I circled: fructose, glucose, dihydroxyacetone, all those. But for this exam, you need to know glucose. So remember, we have the linear form of glucose,
There might be two.
and we have to know the difference between D and L. Right? We go to the carbon furthest away from the carbonyl. That is chiral, and right, that hydroxyl is on the right. Okay, and then so we put in the rest of it. There's just that carbon 3, the hydroxyl is on the left. That's all you really want a quick way of memorizing this. Carbon 3, it's the only one on the left. Hydroxyl. Okay. And what we need to know, right, is that these cyclize. So basically, I'll just draw it like this. That we get tack here. 6 carbon sugars like to make 6 carbon rings. And we said that you can get 2 anomeric forms, right? That can form dependent on which side of the ring it attacks. So how I draw these rings in a real quick way is draw it with your oxygen as a member of that ring. Okay, over here. Here is your carbon 1, 2, 3, 4, 5. Okay. 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 So you just draw your 6-membered ring. Now we call this a pyranose, right? And a 5-membered ring we call a furanose. Does it matter for this exam? Maybe. Right? And we said that anything, okay, on the inside of here, on the right side, goes down. Okay, if you want an easy way of remembering it, or you can just remember glucose because it's the only one. This is how I remember it. When I draw it in this way, it's down, up, down. It alternates. Okay, so down, up, down. And we said this is the anomeric carbon, remember? Because the attack could happen at the top or the bottom. If it's on the same, if the hydroxyl is on the same side as carbon 6, we would call this what form?
Beta.
Beta. Beta, same side. And if it goes down on the other side, we call it the alpha form. So up, down, and because it's going to be our alpha form, it's down. Okay. So we got that. Now, the questions that I usually ask are related to things like what we talked about in class, like glycogen versus, we talked about chitin, right? Or we talked about cellulose, okay? And this is what's amazing to me. You can take glucose, okay? So we have 2 forms of glucose, beta glucose, and it's a D. And we have the alpha glucose form. And if you connect them in different ways, you get different structural arrangements that lead to different functions. So for instance, the beta glucoses, we talked about cellulose. And how is cellulose formed?
Oh, it's
One
like,
for alpha bond?
It's a what?
One for alpha?
yeah, it's the spiral.
1,4-alpha, 4-beta glucose.
Beta? One for
When we,
beta?
when we have a 1,4-beta, that's what cellulose is. Cellulose has what we call 1,4. So let me draw this a little more. If we just wanted to see what the repeating unit looks like, this is what we would see. I'm sorry. So, and then we go up, down, up, down. So if we draw on the Haworth projection of cellulose, it's all 1,4-beta linkages. Okay? And so we would say this is a alpha 1,4 link. Okay? Now what's the difference between chitosan and cellulose? This position right here.
Oh, that was a beta 1,4 link.
Oh yes, I'm sorry. Losing my mind. I was saying the wrong thing. But it's beta, so cellulose is beta, that's what I was drawing. As the arrows distinguish you, right? It's been a long day. Alright, so you guys get that? So what does this do though? We draw it in the Haworth. You don't have to be able to draw this in the chair form, but basically what you get is if you draw this out, okay. Okay, my chair is getting crazy down here. But what you're going to get is you're going to get something like this. Oh. Go back. Go back. Okay. So these are beta 1,4 length. It should come back up. Okay. So what happens is this is really making sheets. So how I draw this is if you know this linkage in Haworth, drawing out the chair and where everything goes is kind of complicated, right? So what you want to know is how can we draw cellulose schematically? It basically does this. It makes this weave structure that stacks on top of one another where there's hydrogen bonding between the sheets. So this structure here, those of you down here, leads to these chair conformations that are very flat and they stack on top of one another kind of in sheets like this. And now what that does is it keeps water from getting in there. Actually, we don't have the enzymes in our digestive tract that could break this down. And many organisms don't. So things like bacteria and stuff do that are found in the guts of things like termites and other things, cows, right, that can actually break down cellulose, but we cannot eat cellulose and actually degrade it. And this all comes from a difference here in a 1,4 linkage versus in the alpha case. And I like drawing the Haworth just because it's easier for me to draw it in this. So here we're drawing now kind of a repeating unit would be like something like this. Again, we would want to draw out glucose in its full. Oops, sorry. Put an H down there. Make it all right. I'll fix it here in a bit. Now, things like starch, for instance, have alpha 1,4 linkages. So we would say this is an alpha. Don't do that to me. So this would be an alpha 1,4 linkage. Now they also have something else too. So we have these linear parts that are alpha linked, but we also have branch points. Who remembers where the branch points occur?
6.
Yep, so we can have 1,6 linkages as our branch points. So here would be our ring. Okay. So we'll go down here. So this would now be an alpha 1,6 linkage. And this is what is found in starch. Glycogen has this, amylose. All of those have not only these 1,4 linkages, but also they can have these 1,6 linkages. Now this is where we get the helices, right? Whereas this conformation makes these chairs that are very flat. This conformation now kinks it and makes helical structures. So if I want to draw this schematically, what we would see is something kind of helical like this, and off of these we would have branch points, okay, with other helical structures coming off. And we call it a whirlwind structure, okay? And that's kind of what's going on here. You know, if we think about just kind of schematically how these are kind of forming chairs, these are forming kind of helical structures. And this is what I want you guys to understand about it. You know, the difference between an alpha link and a beta link can have such a big change on its chemical properties and its physical properties, whereas something like this forms almost impenetrable sheets, okay, and forms the woody materials that make up trees, the plant matter, all of that. Things like potatoes that have starches, their sugars that are on the inside are locked up through alpha 1,4, 1,6 linkages, and this makes the sugar accessible. So, you know, usually I have you draw out things like this, and then you explain, you know, what's the difference? Why does this 1,6, 1,4 linkage, why does the structure that's produced actually make it good for storage of carbohydrates for energy? And we talked about this. Why? Because now we have a large amount of surface area exposed, right? So if we look here, we would say here, lots of exposed sugar. So if you're vigorously working out and you deplete the blood glucose levels, okay, that are needed to keep working your muscle, it's going to start breaking down glycogen, and we need to get it in there fast. We need lots of exposed ends, right, so we can get to more energy. Whereas in cellulose, most of it's all trapped and locked away. It's nothing that can be easily gotten to. It's going to take quite a bit for something to go in here and try to solvate this, because an enzyme usually has to solvate something, right? It's going to take quite a bit to try to get in there and solvate that, because you have to break all these inter and intramolecular hydrogen bonding networks. So it's an excellent material for making up things that you want to be strong and sturdy. But the real difference is one anomeric carbon flipped. Isn't that kind of cool? So, could you draw something like this, and could you kind of explain what I've just said in terms of where does this difference? But they're all made of glucose. This is what's cool. Glucose is the framework for all of them, but depending on whether you have alpha or beta form, it totally changes its properties. All right. What else did we learn in that chapter? That was basically it, huh? Yeah. All right. So we learned about cyclization. We learned terminology like alpha, beta. We learned, things about epimers, things like that. And then we learned about the structure function. That's basically what we got going for that. Anybody have any questions about that?
Yes.
Yes, Dave.
Okay, so why is the 1/6 linkage not possible in the beta form, or why is it possible on the alpha?
So what happens is, it's not to say that you can't have a beta 1/6 linkage. During the process, all of this connecting is controlled by enzymes, and so it depends on what enzymes are present. So in like your starch or potato, when it's storing sugar, it has the enzymes that are both making linear chains, okay, through the alpha 1,4 linkages, and it has another enzyme that's actually putting on branch chains. Whereas if you're talking things like cellulose, you probably don't even have an enzyme that's trying to branch it off.
Okay, so you take the same, like, I just portions of those are exactly the same. Throw in different enzymes. It gets those portions.
Yeah, as long as you have the people like beta versus alpha. And there might even be enzymes that favor.
Right, like maybe an alpha and you want that at some point, that's what the beta one.
Yep. Okay. So let's go on. So that's what you need to know there for chapter 9. Okay. So now what's the question? Math. We like math. We love math.
That's too bad.
Which math? Test 2 math. What was test 2?
Getting the answer.
Okay.
Sequences?
So why don't we just go through? Let's go through each chapter.
You did one sequence.
And let me talk about the main
oh,
points that you should, like 100%, I would have to know.
I like that idea a lot.
Okay. So chapter 2 really is where we start. And that was water, okay? And so what did we need to know from water? We needed to know the structure and properties. So we know that, right, water, H2O, has this kind of general structure. If we want to look at it in 3D, right, we would say, we would put these here, and we might say, okay, on the wedge here coming out, we have a lone pair. On, yes, and then overall, we have an electronegativity difference in the molecule. So we know that, right? And this is what leads to hydrogen bonding networks, right? So we talked about hydrogen bonding networks, yada, 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 right? And we know that every water molecule can bond to 4 other molecules of water. Okay. This leads to what we call the cohesive nature of water. And we see things like ice that actually, as you freeze water, it becomes larger, right? Which is weird for a solid, right? Usually a solid, when you solidify something, you make it smaller. We talked about the main point from this, okay, was to understand those properties. We learned things like dissolves like, okay, and we learned the rational basis for that, right? If you want to interfere with water hydrogen bonding and interacting with itself, what do you have to do? You have to put in something that can also share in a bonding interaction with it. And that was the gist of it. If you can have ionic species do that because of the dipole moment, but if you're going to exchange the hydrogen bonding interactions that water is having with water, you have to incorporate something else. Okay. Now the main thing beyond that we talked about was the ability of water to dissociate. And we had this equation, the Henderson-Hasselbalch equation, right? Which was that the pH equals the pKa plus the log of the concentration of the conjugate base divided by the concentration of the weak acid. And we had to use this, right? And we talked about calculating concentrations of each of these based off of a pH or a pK. Now, here was the main thing that y'all have to remember where I usually see people confused. The pK that you have to use has to fall within the pH range, right? So the pK used has to be typically plus or minus 1, right? So the pK used has to fall plus or minus 1 within the pH we're looking at. Remember that? Because the buffering region is the pKa plus or minus 1. So when we're figuring out what pKa to use, you guys remember this? We say which pK falls within the pH range that we're trying to buffer. You guys all remember that? And okay. And so we talked about that. We talked about the titration curves, which you all love so near and dear to your heart, where we look at pH versus hydroxide equivalents. Okay, now here's why we did all that stupid stuff, okay? Because I know you think it's probably all dumb and stupid, right? Or maybe I shouldn't say it. Here's why we had to do that tedious, work. 2, 4, 6, 8, 10, 12, 14. The whole reason for you doing the titration curve was for you to get a mental picture as to how something dissociates from a molecule. So for instance, let's say I have a peptide. And it's just a tripeptide. We're just going to make an easy one here. Okay? And I'm going to draw it in its fully protonated form. Okay? And I hope you all remember your amino acids too.
Yeah. Sure.
So, how many pKs do we have on this thing?
I don't want to.
1, 2,
1,
3.
2, 3,
Three.
4. 4, right? And you have to remember, right, this is kind of blending in with the other chapters. So we said this has a pK of 2, this one 6, this one 8, and this one 9.5, right? So when we come here, we said, okay, we can draw out the titration curve for this. We put our half equivalence points in between each of these. And then we go in at every half equivalent point, right? And we put in the pKa. So the first one, I'll change the color here to make it observable. So we said that would be the carboxylic acid would be the first one to go. That would be at half an equivalent, 2. Then we would say the next one would be the histidine, right? At 6. So right about there. The next one would be our cysteine here at 8, so at 2.5. You guys remember all this stuff? You learned quite a bit, actually, this semester. At 9.5, we have that final one. And then what we said was buffering is plus or minus 1 of the pK. So what happens? It shoots up as we add hydroxide till it hits a pH of 1. And then we get to that first buffering zone. It's going to gradually increase to the pK. Then when it gets about 1 pK unit above that, so right here's about 3, that's going to be at 1 equivalent. Now what's going to happen? It's going to shoot up to minus 1 of the pK, which would be at 5. So 5, and then we're going to have another buffering region. And then it's going to go from 5 to 7, which will end at the second equivalent. And then it's just going to keep on going up linearly, right? Because cysteines, it starts at 7. So we're not going to see much of a dip there. And so we're going to go from that 8 over up to 9, which is going to overlap with this. And then we're going to keep going from 9.5 to 10.5, and it's going to go up. You guys remember all this?
Yes.
Now, the important thing about this is for each of these pKa ranges, this is telling you, okay, if we're below the pH of a pK, what do we know? That it's going to be in its protonated form, right? So here we would say, oh, this is where this species, this functional group here is neutral, right? Then when we get above this pK point, what's going to happen? It becomes negative, right? And then when we go up here, on this side of the second pK, okay, we're talking about histidine here. What is it? It's positive, right? Yeah, on this side, histidine is going to be what? Positive. Then when we get above the pK, what happens? pH, it becomes neutral. And this is what it's supposed to give you an idea of, okay, if I'm on this side, it's protonated. If I'm on this side, it's unprotonated. And from that, knowing the functional group, you can tell what the charge is going to be. Okay. You guys remember all this stuff? And then we go up there, this is 0, then it becomes negative, and then this becomes positive, and then it becomes neutral. I'm just looking at the individual functional groups. So this gives you kind of a mental picture of how this whole dynamic is going. Does that make sense? Okay. So now here's the question. Okay, where do we reach the PI for this thing, right? So starting off all the way down here, right? We're starting at what?
After the second one, +2.
If we're fully protonated. And I'm showing you the fully protonated.
+2.
+2. +2, okay, so we start here, we get to here, and what happens? It subtracts one of those, right? We get one of those, this one became negative, so what did we at here?
+1.
+1, right? So this is a quick way of, so we're at +1 here. Okay.
It's still +2.
Now we go up here.
+2.
So, see? +2, we put in a negative, right? So it's +1 at this stage. Talking about this molecule. We get past its first pK. Then we go up here, past this one, what happens? This becomes neutral. So now what is the charge?
0.
0. So we found our isoelectric point. It's the pKa between, right, where it actually makes 0. You add the 2 pKs on either side of that, right, and add them together, divide by 2. We got our pK, our PI, right? So it would have to be in between this one and this one, right?
Oh, you're saying you lost that positive charge to get to the neutral?
Yes.
Okay.
Yep. So we would take, in this case, oops, sorry, 6 plus 8 divided by 2. So 7 makes it easy. Isn't that cool? So it's just a way, if you have this mental picture in your mind, you don't even have to draw every protonation state. You know, that's how I originally did it, right? You had to draw every single one. But you could quickly come up with an idea from your knowledge of how each one works. Okay. So could you draw this? Could you draw titration curves? Could you, draw out the structures of these molecules? Could you do a quick back of the envelope, basically, calculation? And I feel like that's useful information for you. And I know some of you are like, I'm never going to do this. Well, you might. Okay. You never know. So let's say I said, what are the predominant species and relative amounts at pH, let's do 8.5.
Ooh.
All right, so what are the two species? Okay, well, I know 8.5 is above the pK for cysteine, so I got to draw the structure, okay, of it before it's deprotonated. So we're still going to have this, H3, and okay. Oh, okay. So one will be the protonated form of cysteine, right? And in both forms, histidine is going to be deprotonated now. Okay, so we've deprotonated that because we know that we're above, right? It's pK, well above it, so we're going to be like that. The two forms are going to be the protonated form and the unprotonated form. And I like to show my electrons. You don't necessarily have to. All right. So, and then you all can. Oh, and I need to deprotonate. You guys let me do all kinds of stuff up here. Yes, you tell me later. That math you didn't make any sense.
We're testing.
Yeah,
I still want to see on top of this.
that's okay. All right, there we go. So those are your two forms. Okay, for the pH, we would say, okay, pH is 8.5 equals, we find the pK that's within that vicinity, the closest one at 8.5. The closest one would be pK 8. Okay, so I would say right? We can solve for the ratio, and you guys feel comfortable with doing problems like that, right? Yeah, and even if I said, well, what's the, you could even take this further. If I gave a concentration, like we did way back when, you could come up with another equation, and you guys remember all that kind of stuff?
Yeah.
Yeah, that's good. Right? You make up two equations. Well, let's say I said it was 50 millimolar of peptide. You guys remember this? And then what we would do is set up another equation. We would say A minus plus HA equals 50 millimolar. Remember that? And from that we could solve for this equation. We would solve the ratio. And then we would rewrite this equation and write it in terms of the other. You guys remember that from algebra? The best one to do is to solve this first. So we would subtract, you get 10 to the 0.5 equals A minus over HA. So what I did here is I just took, right, I subtracted this over here, got 0.5. To get rid of a log, remember you take 10 to that power. So that's why I said 10 to the 0.5. This is our ratio. Okay. Then what is 10 to the 5th? 10 to the 0.5, is that 10 to the 3.16. So,
Yeah, 3.16.
yeah, I got 3.16. So we say A minus over HA equals 3.16. Now what we would do, right, is if we take this, bring the concentration of weak acid up here, we get the conjugate base equals 3.16 HA. We plug that in, right, for the conjugate base, right? And then we can solve then for our constant. Remember all that? Yeah. Go back over and look over that because that's fair game. That's what we're doing when we're making buffers in labs and stuff. Okay. So we all cool with that? So that was chapter 2, which spills over into chapter 5 and 4 and all of those. Okay. Chapter 3 was thermodynamics, right? We don't have to really go over all this, but we have a few general equations that you have to remember. Delta G equals delta H minus delta ST. The one we focused on more, though, was the one that basically describes chemical equilibrium, right? We said that delta G not equal, oops, sorry. I don't want to say delta G not just yet. We said that the delta G of any reaction, okay, equals delta G naught prime. Do you remember what this means? This is under the standard conditions, right? Equilibrium, standard conditions, plus RT ln Q. Remember that? And this is for chemical equilibrium. Remember, Q is the prevalent conditions. Is this working okay, or am I taking too much time on this stuff? Okay. 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 So remember, Q is this. So if we had an equation A plus B, C plus D, we know that Q, right, is products. So concentration C to the C power times D to the D power, however many products, the products of those products divided by the products of the reactants. This one was one that tripped up some people on the exam, as I recall. Right? Depending on the conditions, what do? We modify from the standard state because we're not at standard state conditions. We can get a new Gibbs free energy, right? So we would say if we have an excess of, say, reactants, right, away from equilibrium, what's going to happen? Delta G is going to get more towards the negative, right? Because the smaller this, you know, our log Q is going to shift things toward equilibrium. Okay. Does everybody remember this? Now, if we're at equilibrium, delta G equals 0. Remember that? So we get this equation, delta G naught prime equals minus RT ln of the equilibrium constant KEQ. So if I provide a KEQ, you could calculate the Gibbs free energy under standard conditions. And if I gave you a chemical reaction with the conditions, you could plug that into this equation and you could tell me whether it's exergonic or endergonic. Remember that? Exergonic is where you have an overall negative delta G. And what does exergonic mean? Is it favorable, spontaneous?
Yeah.
Yes. Yes.
Okay.
Endergonic would be a positive delta G, right? And that is non-spontaneous, or the way I like to think of it better is it's not favorable. Okay? So when we talked about thermodynamics, this was the brunt of it. We talked about how reactions can be swayed and shifted depending on conditions, right? So even if you have under standard conditions a positive delta G naught prime, Does that mean it will never happen? No, because if the conditions change, we can shift it around. And all of biochemistry really plays with that principle. That if we control the conditions, the concentrations, we can push a reaction maybe that's normally not spontaneous. We can drive it and make it spontaneous by keeping, for instance, products low and reactants high in the cell. So this will come into next semester. Yes?
Okay, just like making sure that temperature can also be used to sway the
Yes.
positivity slash
Yes, but in us, it's going to be 37 degrees Celsius,
yeah.
which is
So like if,
298.
so like if in standard conditions it's negative delta G, but in the cell it's at like a higher temperature,
Yes,
it could be positive.
yes, this is what swings. It's either temperature or Q.
Okay.
Remember, this is an important point.
Will we give them that and solve that?
I will give you R
Okay.
and things like that, but T, remember, is in Kelvin. And we take degrees C plus 273.15, that gives us our amount in K. So in the cell, you know, we're adding 37, so it's 210 K. It's pretty much constant in our cells, but it's higher than normal, 25 degrees. So does that all make sense? So this is how I think of it. This is a state function. Delta G naught prime is our reference state, and we can change the free energy that's available dependent on temperature and the conditions. So that's how we shift it around. Okay, I think we spent enough time on that. But that's mostly what I was talking about, understanding all those things, thermodynamics, negative delta G's, things like that. Are we okay with that for chapter 3?
That's the extra credit question, yes?
Yes, sir. Maybe. Chapter 4, we talked about nucleotides, so nucleic acid. Do you remember all the structures? Can you draw all the things? Do you remember the difference between a nucleotide and a nucleoside? You guys all remember this? So we said that a nucleoside is where you take a ribose sugar. Right? We have the purines and the pyrimidines. Purine, shorter name, bigger molecule, bigger base, right? They all have this basic structure. Adenosine, right, is the first one we learned. A, right? Younger in the alphabet, A is the first one, so it has hair on top of the head. That's why we have the NH2 on top. You know, as we age, though, so if we're talking about ribonucleic acids, so having ribose, we would call this a nucleoside, okay? And this would be adenosine. The other one, right, guanosine, is where it ages, right? It's older in the alphabet, so what happens? We have the same basic structure. NH2, the hair falls off the head, lands on the back, the head gets oxygenated, right?
Oh.
Let's ask him, do you have more guanosine, Dr. Smee? Don't tell him I said that. All right. Guanosine. All right. All right. So we had those two. Those are the purines. Yeah, that's an N up there. Yeah. Or I could just be drawing it, and I don't know. They know how to spell it, and I just make it messy. You guys just give me a credit. No, I'm gonna do that. Okay, so these are nucleosides before you actually activate them with phosphates. They become nucleotides when you start to remove this and you put on actual phosphates. So we would say this, And I'm just going to make them both negative. That's depending on pH. Okay. Okay, it's very off from where I'm trying to draw. But we would say as you start to add phosphates, I'm going to put an H there, we would call the red ones nucleotides to distinguish them from nucleosides. So we would call this adenosine 5' monophosphate. You guys remember that? We call each of these positions, we give it a number, and we call it a prime to distinguish it from the ring. So you guys remember drawing all those? And the hair thing also works for the other ones, right? The pyrimidines. So again, we have the same sugar. So you do have to know ribose, right? Because we found that here. The structure is slightly changed. It has a singular kind of aromatic structure. We have here cytosine. Okay, and what is this? This is younger in the alphabet, so what's going to be on top? The hair. And over here we have oxygen already here. Okay. Oops, I'm sorry. I screwed up there. That would make that nitrogen really positively charged. So I would rather draw it that. Okay. And then we have the other one, uracil, if we're talking about RNA.
So you got the double bond with the nitrogen on the right hand side?
What's
Okay.
What's that? We good? Oh, yes, yes, we would have that. Yes, sorry about that. Yes, you are correct. Okay. Then what happens, you age, you know, the hair falls off the top of the head, but on this case, there's already an oxygen there. So we get uracil. Okay. What's that? We're an RNA, so there should be an OH.
Oh, so we're all talking.
Yep. Now, if we are talking about DNA, right, thymine, what we would do there is we would take that off and put that out there. Remember, there's a methyl group. So, to distinguish thymine and DNA. But then, remember, all DNA is just removing those on them. So, you guys are good with that. And then how they're connected. You guys remember how they're connected through phosphodiester linkages. So it's a 3 to 5 prime. Okay. So I'm just going to draw real quick because I know this is so exciting for you all. So remember we have. I'm going to draw notch from there, but it's not the best place. Let's drop from the 3 prime this way. And I'm just going to draw it like that to say, okay, we have base 1, base 2. Okay. I'm just going to draw that. If it's RNA, all right, we have that there. If it's DNA, right, we remove that. DNA. Okay. And then what kinds of things? So you should be able to draw out the structure. Remember, there's a directionality. We would say this is the 5' end and the 3' end. And if I gave you a sequence that was A, G, T, A, you would draw out a structure that would have the same backbone, but at the different positions, you would have A starting first, right? Because it always goes 5' to 3'. Then the next one would have G, T, and so on and so forth. You all remember that?
Do we have to know how they line up?
No, we don't have to do, so you have to know that too. So you have to know that adenosine matches with uracil or thymine in the case of DNA. You have to know that guanine base pairs with cytosine, and you got to be able to draw that interaction. I will not have you draw polymers of them and draw them, because the 3D, they actually set kind of linear and flat when they're base pairing, and it's hard to do that on a piece of paper. So I don't think you
So
do
you're
that.
saying you'd only have us draw the A, the D, the T, and the A, not the complementary? Or
Yeah, I wouldn't have you draw two strands and complementary strands base pairing. I would have you maybe draw the base pairing between two nucleotides on
Okay.
paper, but
Like
other-
just draw the A and the T.
Yeah.
And show how they interact.
Yeah. And then we also, I think the question that's fine is asking something about the difference between the structures that conform and the stability of RNA versus DNA. Okay? So for instance, DNA, what's the most predominant form that we find? The B DNA, right? There's the A form, B form, and Z form. B form is common for DNA. Z form is very controversial. They believe it's there, but it's not very predominant. A form is found in both DNA and RNA, okay? And it's just a slight difference in the sugar conformation, okay, and the base conformations. But why can't RNA? RNA doesn't form B form. Do you know why?
because the steric hindrance on the
Yeah, steric hindrance on of the hydroxyl. Steric. So it's always that answer, right? Sterics until it's not sterics, until it's something else. But also the stability. Remember, we talked about the stability of RNA versus DNA, which is really important. Why is RNA more or less stable? Sorry.
Because it'll be
Reactive
like an
hydroxyl.
on itself.
Yep. So if we have here, let me erase this, and we have some sort of base here that can grab this. Where could a base come from? There's bases all over in here. So if you have a aspartic acid that's in the vicinity, and then this would go here, right? Attack that, and then that could break. Whereas in DNA, what cannot happen? We can't attack there. It's not going to happen because there's no hydroxyl there. So that's why DNA is more stable than RNA. Okay, so we learned all that stuff. And that was pretty straightforward. RNA, remember, has more elaborate structures. It folds more so. Probably like a protein, we can get unique structures. It can also base pair and do all that. It can form double helixes, but there are slight differences, and you want to know those and recall. Okay, so that was chapter 4. Chapter 5, amino acids, right? And you need to remember all the amino acids, how to draw them. You need to know all the pKa's. Remember, every single amino acid has the same basic structure. I'm drawing this in the most protonated form. We have an alpha carbon to which is attached an amino group, a carboxylic acid, some R group, and a hydrogen, okay? Now, the way to remember your amino acids, hopefully you haven't forgot, besides glycine, everything is just a derivative of alanine. So we would say, okay, glycine is the only achiral. So it's, and they're all L configuration, right? It's the only achiral one. Everything else is a derivative of alanine. So we all start with alanine, and then we go from there. I group them together according to their properties. That's how I memorize them. Do we need to, we probably don't need to go over all that, because that should be our
Can you go over the PKs or?
yeah. So how we remember PKs, 2, 4, 6, 8, 10, 12. PK 2 is the alpha carboxy, right? That's that group there. That's what we remember for them. Within the 4, we have aspartic acid, which is 3.8, and glutamate, which is 4.2. 6, we have histidines. 8, we have cysteines. At 10, we have 3. We have one at 9.5, which is the alpha amino group. At 10, we have tyrosine. And at 10.5, we have lysine. 12, actually 12.5, we have arginine. So that's how you remember those. 2, 4, 6, 8, 10, 12. And you can just bin them together.
But we only need to use those 12 so under the curve. Don't need to use this ones on the side. The 3.8 and all that other, just put 2. Like you gave us a if you gave an asparagic acid and a glutamate.
Yeah?
Just put 4 for both of them?
No, no, you have to know the right ones.
3.8
I
and
just,
4.2.
I'm showing you how to bin them. To memorize.
Let me check and give you a
All right. Then so we learned about that. We do titration curves. We've already done that. We talked about PI. To have a PI, remember, you have to have Zwitter ion character, right? There's both a positive functional group and a negative one on the same molecule. So in the example that I gave you in homework 1, or not homework 1, in exam 1, I gave you something that did not have a PI, which confused someone. If it starts out neutral and never has a positive and a negative charge, it has no PI. But anyway, okay. Then we learned about how they get connected together, right? So we form a peptide bond by what we call a dehydration reaction. So here's R2. What happens? This attacks here. Okay. This breaks off, grabs that. Okay. Those electrons stay there. Water leaves, and we form the oh, I'm not even drawing down there, but it's drawing down there. NH2, R1. So this should be, you know, at this point, pretty common knowledge, I hope. And so this is how the proteins are built from amino acids, right? And we start connecting them together. Water is lost, so this is lost. You should know this mechanism kind of for how this forms. And we always draw it in this fashion where the hydrogen and the carbonyl oxygen are on opposite sides. And we have to know something about the chemical nature of this peptide bond. You'd have to be able to explain that it's planar. Why? It actually has some sp2, okay, nature, because we have this lone pair of electrons that can be donated here. So there are 2 forms, right? We have resonance shared. We have something like this and something like that. I'm just going to leave it that way. So in reality, right, we have these different forms. And so why do we draw it this way? It's actually planar and flat because it has double bond character. And why is it that we always draw these on opposite sides? Sterics, yes. So just like everything in chemistry, right? The trans configuration, where these are on opposite sides, right, is more stable. Okay? So, right, we have double bond character. Okay. Does anybody have any questions about any of that? We all remember that, right? And then we have to be able to what? Draw a peptide if I tell you to draw a peptide. Okay? So if I say draw the peptide, let's see, CYQRS, we always know, oops, that this is the N terminus, right, and C terminus. So we all know, right, that this is the N terminal side, and it goes to the C terminal side. Okay, so what do? Quick way of doing it. How many amino acids are there? 1, 2, 3, 4, 5. Draw on your backbone first. Remember, we alternate the alpha carbon going up and down because of that rigid amide peptide bond. I got now 1, 2, 3, 4. We need one more. And we always have to be responsible and look at what the pH is. I'm going to say pH 4 so that we put the right protonation states on. Okay. The N terminus would be positively charged. Cysteine, we got our wedge. You can go ahead and start putting in, remember your dashes and wedges alternate because we flip the stereochem because of the bond. So we have cysteine. pH is below the pK, so it's protonated. We have tyrosine. That's a 6-membered ring. Okay. Okay, pH is below the pK, so it's protonated. Q is glutamine. And R is arginine. Every amino-containing side chain has 4 carbons. Okay, but arginine has 1, 2, 3. Then it has a nitrogen. Then its 4th carbon, which is attached to 2 other nitrogens. One of which is double bonded. One of which is not. At pH 4, this is a pK of 12. This will be protonated. Positive charges on the double bonded one. And serine. Oops, sorry, that's cysteine. Sorry. OH. pK of this is 2. pH is above it, so it will be perfect. There we go. See, you can do it pretty quick. And then hopefully you can answer things like, where are the phi and psi bonds, right? That's where structure comes from. Phi and psi bonds. So here's a phi. Maybe you can read directions too. If I say 1 phi and psi bond or 2 phi and psi bonds.
Overzealous about
Yes.
psi bonds.
No.
I
No, no, no.
know. Yeah.
It's like made a big point of that too. Just put however many you need.
Right? And depending on the phi and psi bonds, what do your repeating phi and psi bonds give you?
Flexibility.
No, well they can, but repeating where the phi and psi angles repeat. What does that lead to? Secondary structure. Okay, so along segments. And that leads into protein structure, right? We have different kinds of protein structure. We have primary, secondary, tertiary, quaternary. The linear sequence of amino acids is primary. Secondary, local repeating patterns of structure within a polypeptide. Tertiary, 3D structure of a single polypeptide. And quaternary, overall 3D structure of several polypeptides. Okay. We can also answer questions about Chandrian thoughts, right?
Nah.
Yes. So, yes.
In my notes, I have that lysine has one more carbon for the first nitrogen. I don't know if
It's not lysine,
or
it's arginine.
arginine.
Should have 3. 1, 2, 3, nitrogen, 4. Should be 4 carbons. 1 from the alanine. That's right. 1, 2, 3, 4. And you want corrected notes. So cool. So remember what allows certain structures to form, right? Why do we see basically alpha helices and beta sheets, and we see variations of those, but why don't we see other things?
Allowable bond angles. Because of steric hindrance.
Steric hindrance, right? Those position the R groups far apart from one another. Now, what's the differences between an alpha helical structure and a beta sheet structure, besides fine sine angles? What's one of the differences?
The hydrogen bonding.
Hydrogen bonding. In an alpha helix, what's the difference? It's within the structure itself. Beta sheets, what do they require? Two segments, right? So one's kind of self-contained, and one requires another part of the protein. All right. So we learned all about that. That's basically chapter 5-ish, and then chapter 6. What's chapter 6 is structure, right? And yes. Oh, and we need things like chaperones, folding landscape theory, right? We love that. Protein folding landscape theory. So how do we think of protein folding? Minimizing energy, right? And so we think of it, according to this theory, as a funnel, right, where we have entropy, kind of a pseudo 3D, around the kind of x-axis, going down, right, so higher delta G's, up, okay, and then we go to the lowest energy state down here. We would call this the native state, okay? And that's how we think of it, right? That the interactions are coming together. We're getting an overall more stable structure, even though it's becoming lower entropy, right? It's becoming more ordered. And that's how we think about it. Now, this is an idealized one. Remember, there's a contour to it. They can actually have intermediate states. This is kind of our ideal form. So you guys could explain that. Y'all remember that?
Do you want the ideal form or do you want the other one?
I don't care. Well, know both. Know that this is an ideal form. There can be other ways. And then how does that enter things like chaperones, right? They help fold by basically guiding back onto a proper folding path. And this is in, I think I gave you a question on this in the exam 2 or whatever.
Is there any particular reason why it's delta G prime?
I'm just showing that this is at standard state, so it doesn't have to be. So I'll just circle that.
That's gonna be
Maybe. I'll take it off. We'll just say delta G. Because it is concentration dependent and what surrounds going to influence it. All right, so what do we know? A lot. Okay, so what else could I ask from that? Those are good. Those are good questions. Okay, now we go chapter 7. Protein function. Yeah? So here's the math things that we were talking about, right? We said that proteins function just like people through interactions. So we have a protein plus a ligand, and we said that they interact through a protein-ligand interaction. There are rate constants for this, K1, K minus 1. And I'm not going to have you derive. Yes, sir?
But do we have to know like techniques, purification techniques?
Oh, yes. Yes, we know.
Okay, thanks for sharing.
Yeah, thank you. But before we do that, let's talk about function. Okay. Yeah. Cool. So, for instance, we said that how do we actually evaluate this? We look at theta, which is the fraction of sites bound, right? Which we said is equal to the ligand concentration divided by the ligand concentration plus a KD. Remember, KD is K minus 1 divided by K1. So it's those terms wrapped up together. The lower the KD, the higher the affinity of a ligand or a protein for itself. Okay, so we all remember that. We can look at a plot. It would look like this, where this is going to 1. Now, this is what people usually mess up on. They usually confuse Michaelis-Menten plots with ligand binding plots. They look identical. Why? Because a protein, an enzyme is a protein. It binds to its ligand, its substrate, but then it does an extra thing. It converts it to a new chemical, right? So we don't get to see how much is bound because as soon as it binds, what happens? It changes it into something else and lets go. So our plots, remember, theta is a never-changing thing. You can only have a certain fraction bound. Now, when we go to enzymes and we start talking about BO, that's a changing entity depending on what? How many enzymes we have present. It's going to influence the rate. So do not confuse those two. Where do people confuse it? In figuring out the KD, right? The KD is found at half the binding sites bound, right? So this is a KD. We always go to 0.5. So always, and for good measure, always go to 0.5 theta. Okay? For KD. Okay? Remember, Vmax changes in your enzyme kinetics plots, so you're looking for a different half Vmax when we're talking enzymes, right? This, never ever look at how high this goes. Always go to 0.5. Does not matter where this reaches. You always go to 0.5 theta. And I'll be happy grading your papers because I can just go on and cruise through it. All right. We doing okay? All right. So now the question was on purification, right, and stuff. Before I go on to the rest of this, yes, purification is fair game. Techniques. Sequencing is your favorite one of all. Yes.
Can you tell us if it?
Yeah, can you give us the list of those 8 different things then?
I can give you.
All
So,
the
well, let's.
On the test.
Can't give you any list of anything.
Like no card?
No, heck no. You're a lean, mean biochemist machine, man. Come on. You can figure this stuff out. You got it, John.
I just missed it.
All right, so things like
A 16-point test?
salting out. And those of you who are in the lab, you got to
Can't
step
miss that
ahead
one.
of this because you've done it, right? I think you've done it. You were there in person. I don't know much. Anyway.
Oh, we did it, but we didn't get good results.
Well, that's another thing. Salting out, centrifugation. Main ones are like ion exchange chromatography, size exclusion chromatography. What comes out first on size exclusion chromatography?
Largest.
Large.
Largest first. It took the highway. It took the highway. Okay, that's why. What else did we talk about? We talked about affinity, chromatography. Those would mainly be, ion exchange, right? PI, right? Okay. You got to know how things elute based on PI. So we have two kinds, right? Cationic exchange, high PIs elute last. Okay, right? You want to look at anionic? Yep. Low PI, I should say, not pH, PI last. So remember those orders. Things like techniques would be like definitions, like what is it? What is electrospray, ESI, electrospray ionization mass spec versus MALDI-TOF, matrix-assisted laser desorption ionization, time of flight mass spectrometry.
Are you
You
just
ever
making
those?
this up?
Yes.
No, I'm not making this shit up. I said a swear word. That wasn't right. Yeah, I just come in and say something. You must know what he's talking about.
What was the first one?
I can't remember mass spec.
So one is spray it out.
Yeah.
The other one is use a laser. So
Chromatography
what's
is
the
cold
difference?
room.
One you spray it out through an aerosol, the other you use a laser. But little definitions. Now here's kind of what I like to do. So let's get through the rest here and I'll go. So those are mainly what we talked about there. Now the sequencing stuff, here's what we need to remember. Edmond degradation gives you N term to C term sequencing. So if I said Edmond degradation gave you a sequence A, C, Y, that means that this is the N terminus of the peptide, and this would be its C terminus. It's just giving you the exact sequence of whatever peptide you're giving. Now, if we start hacking something apart and we get two sequences, one is for one peptide, the other is for the other peptide, okay? But that's all Edmond degradation is. It's just telling you the sequence. N to C terminus. Okay, now the things that we need to remember are where things cut. So, chymotrypsin cuts at tyrosines, tryptophans, and phenylalanines on the C term, right? Aromatics. Trypsin cuts at lysines and arginines. C term, right? Cyanogen bromide. Methionine, C term. And aspartate protease. So D, C, N term. Sorry, it's in the thing. I'm just used to saying C so much. Anything else? Carboxypeptidase. Yeah. Peptidase. It's the C terminal residue. C term residue. Whatever's on the end, that's what's happening. So it does cut on the N terminal side of the last residue. Okay. So it just tells you the N. It tells you the C terminal residue. For sequencing, remember what does Elman's reagent?
Nitrogen?
Elvin's
Cysteine,
reagent gives you
right?
cysteine, right?
Cysteine tells you have cysteine present. What does
Together.
fluorescence tell you?
W, whatever W is.
Yeah, whatever W is.
Tryptophan, dude.
It's not looking good, Jeremy, man.
Whatever W is.
Whatever that is. Okay, why does this matter? Because HCl, right, concentrated HCl destroys those. And so we always take the intact peptide to see if those things were there.
And it switches the.
Yeah. And then also, HCl converts asparagines and glutamines to aspartic acids and glutamic acids and NH4+. Right? So we know if we see NH4+, we actually had one of these there before. You guys remember all that? Ringing a bell?
The belly.
Enough the bell's been rung. All right. So all that stuff. Now I can switch things up a little bit, and I'll show you how I could modify a question. You have to know these cuts, but not necessarily just for sequencing. I could have you know it for a different kind of problem, and I'll show you. So is everybody okay with that? No, good. Good. I hope in my evaluations you put something about caring, attentive, you know, like he really does care. When we don't get it, he just nods and continues on. Chapter 7. Now the other thing in chapter 7 we did was hemoglobin and myoglobin. Yep, cooperativity. So tetramer, monomer. And all these terms that you didn't know before, hopefully it's kind of just sunk in and you just know them, right? And we said that this right has cooperativity and this no. And we said why? Quaternary structure. And you have to have communication between between subunits. So one of the things that was on the last one was provide similarities and differences. You remember that? I think I gave you that problem on there, didn't I? Yeah. Maybe I should think of something new. Huh? They all have what in common? What do you-
Same, the hemoglobin.
They all have heme,
What's it, peripheral?
heme, and iron. They all, the porphyrin. They all have a distal and proximal histidine.
Also beautiful.
Do you remember what the proximal histidine, right? What they do, right? What else is similar?
Carry oxygen.
They carry oxygen, yep. Carry O2. Differences, we've already kind of given one. Quaternary structure. Now, another one common was their tertiary structures were identical, right?
Yeah.
Quaternary structure, sequence, right? There's only like 27 amino acids the same. That means the rest of the 300 or 400 amino acids are different. So that's pretty amazing. So I should put only. Sequence is different. Quaternary structure is different. What they do, how they deliver oxygen, O2 long range versus local, right? Hemoglobin's the semi-truck, and then the myoglobin is the little transport car, compact, yep.
U-Haul.
I don't know that it's U-Haul size compact, but anyways, anything else worth cooperativity? Cooperativity.
Wasn't the source of them different? The source of them were different?
You mean where they're at? Yeah,
When they're
what
made and manufactured?
that could be. I would be okay with that. Okay, and then also the other thing you need to know is the mechanism of hemoglobin cooperativity. Ready?
Oh, that's not the whole right.
T to R, yeah.
With the
Pucker.
head being pulled in, learning how to play in the cooperativity.
Yep. Remember that? The molecular basis. This is what the whole class is, right? The molecular basis. And it's all coming back, right? You don't even need to study, right? It's all there.
As long as your test is exactly what you wrote.
When you say draw, like,
And we can even talk about thermodynamics, right? Why does it go from T to R? As oxygen concentration gets up, what happens? The R state becomes more stable, right? There's that transition in delta G, right? So we got to think about these structural changes in terms of energy too. Why would it even want to do that? Well, as the oxygen concentration gets high enough, it's going to favor the more stable structure. So I like that kind of stuff. It's great. It's not that bad. It's really not. Okay. Yeah, I know. I teach it, so you write the exam. What the heck you got to worry about, right? You get a PhD? Yes. So we good? So that was, there was the other ones, you know, we talked about in passing antibodies, which you studied for on the last exam maybe. Antibodies is another class, and myosin.
Are you gonna way more heavily on like the others, or it's gonna be all equal?
It's random. I just get a dartboard and kind of, this chapter, what do we do?
well, we have 5 sequencing problems.
Now, you get an extra half an hour with the exam, so. Okay, and then the next one is chapter 8, which we spent a lot of time on. And we're tested on, so I don't feel like I really need to go over it unless you have some specific question that you were like, I don't, so. Right, so we need to be able to do things like determine Vmax, Kcat, Km, catalytic efficiency. Okay? Remember, Kcat is Vmax divided by the total. Catalytic efficiency is Kcat divided by Km.
What are the units of that wind unit supposed to be?
Well, if Kcat's in per unit time, so per second, this would be in terms of per second, and Km is usually in concentration. If it's in molarity, then it'll be per molarity, per millimolarity. Those are the units. Kcats are usually in terms of per second, per minute. Okay. Remembering too that the enzyme concentration has to have the same concentration units as Vmax. So if your Vmax, for instance, is in terms of millimolarity per second, then your E total also has to be in terms of millimolarity, so we can cancel those out. So, I mean, everything from the last exam. I covered the whole thing, basically. You saw it on the, I gave you a question on every kind of question you can get for enzymes, because that's all we did. So, no.
I say that.
What's that?
Especially one.
I'm about 2/3 of the way done, so I have to. I just got to get it. I mean, okay, so it's, now that means inhibition too. Inhibition.
Oh.
Take a look. Some of you, most of you did really good on the kinetics parts of the exam, from what I've seen so far. Everybody was pretty much nailed. The only thing I knocked people on was units. People keep on making me upset. What was your question?
You were mentioning a second ago about sequencing, but you could ask it in a different
Oh
way.
yeah, I'll do that. So we're good with all the kinetics. We're good with explaining things like, you know, steady state, all that stuff. You can look at my answer key. Now, things like mechanisms. Mechanisms are still there, okay? We had 2 that you need to know. Now, here's a cool question that I could do. I could do this. I could say, draw the peptide. Let's see. A, K, N, W. And show the mechanism for its cleavage by trypsin. Alright, cool. I like this. So we know trypsin is what kind of protease? Serine protease. So if you don't see something that says lysozyme, you can assume it's a serine protease. Okay? Okay, so what do we have to do? Okay, I've got to draw this peptide up first. So we're pulling stuff from chapter, 4. Did somebody have a hand raised? Oops, I'm sorry. I'm going crazy. So we had what, 4 in it? Right? 1, 2, 3, 4. Okay. And we'll assume neutral pH, okay? And then so we have NH2, or NH3, I'm sorry. And so alanine, lysine, right? 1, 2, 3, 4, NH3 plus, asparagine. Is everything looking okay? Oh, yep, that's tryptophan. All right. Let's all go have a drink. Okay. Now, of water. Yes. Okay. So we know something about trypsin. It cuts after what? Lysines and arginines. And hey, we got one of those. Right there. So we're going to cut on the C-terminal side. And so how do we, oops, sorry. How do we do this? Well, we have this catalytic triad here. So we have our serine here. And we have our,
How'd you know it was going to be the second one again?
because I know trypsin cuts after lysines, and I know that it cuts on the C-terminal side of lysine. Lysine, yep. See? Okay. Sorry. Okay. Now we can just put in what I refer to as grandma here. Aspartate is grandma. She sits in the corner. We know she's there, but we just kind of let her do her thing. Right? Is that what you do at Christmas time or whatever? You just leave her over there? Oh, no. Yes. We know that's hard. My grandma's dead. Well, it's okay. It's okay. She's not in the corner anymore. Oh, that's bad. Oh, it's okay. All right, so we have to attack here.
I don't know, man.
Yes, not all your opinions are changing, right? Can I have that evaluation?
I think it's
All right, so that grabs that, and then this attacks here. That's gonna pop up there, right? And then we're gonna go to the next stage of this. Now, do you think I would have you draw out the entire thing?
Yeah.
Every step?
You hear Christian, yay.
Oops, I'm sorry. That's just
So all 4 steps.
Yep, 2 points off. You guys have seen my grading scheme, right? Because 1 is the lowest number, man. gotta have it in pairs. Well, 0 doesn't know anything. It doesn't know if it's alone or.
Well, that makes it, should be used more often.
Oh, I see.
So it learns.
Don't give me this.
Can we just?
Oh, no.
What's it on the-
Because it's not good if people are already saying, oh, no.
So you could.
So, we got Siri here. Yeah, looking at it, I probably won't have you draw up the whole thing. You have to read the question carefully.
Yeah, this is the same class, it's not a free class.
I'm fine with it.
You would think so. But, okay, so then all we do is to keep all the clutter, you know, we can put in all the side chains later. This is the easy way of doing it, right? We just put it, okay, so what's gonna happen? This is gonna pop down here. Okay. Okay. Then this is gonna break, and it's gonna grab that, and the electrons flow to the sink, right? The nitrogen, positive nitrogen sink. And then what happens? We go down, we get something that leaves here. NH2. Oops. Sorry. And then we could put our things in there later. That would be 5D. And this would be our all right, there we go. There's
On
that.
the exam, like, would we get points off if we didn't draw them like all the way out?
It depends on what the directions say.
Okay,
Make it as
so
broad as
if the directions don't explicitly say I can't do this, I can like put their
If it doesn't
abbreviations in there.
specifically say, I want to see all functional side chains, then you could do a shorthand.
Because
Yeah, if it,
I know myself, I ain't got room to draw.
Draw one, but chemistry. You draw your product. Oh,
and so y'all probably hopefully studied this.
I
Sorry.
didn't think I could.
And
Yeah, well.
see what I'll do is keep talking.
I need those 2 points.
Okay, and so then we have our nitrogen. Okay. Now this is reformed, right? And then what happens? Water comes in. Our friend, happy Mr. Water. He is promiscuous. Above all else, he likes to be promiscuous. He's like a cat. Water's kind of like a cat. So that attacks there. We form another tetrahedral intermediate. You guys get it? You want me to keep going, finish it up?
Keep going.
Keep going. We're going the distance.
We have to use
We're
our
going for
you have to-
speed.
Yep.
If you're in my lab, you know that I see this in the lab sometimes. So I'm trying to get everybody out of the lab.
Where'd you guys come with?
What,
Oh,
the last
and
one?
this goes up.
I'm gonna do that.
Well,
Oh, no. Is Dr. Black singing in class?
I think it's due for the last day on Thursday. so-
Dr. Black singing in the lab?
On Thursday.
Dr. Black, is he singing in the lab?
What about?
Is he singing it?
Not.
Probably,
Maybe
actually,
not getting your
I'll
money's
start it tomorrow and finish it.
Go up and demand it. He's singing it.
see how bad I was that.
I'm just messing with
So
you.
much better. gotta make those results make sense. Which I have no idea.
All right, and then basically that will fall off. Are you overwhelmed yet?
Yeah.
It's not too bad.
I'm gonna go have a panic attack.
I mean, if this problem is like the entire test.
I mean, that was like 100 points on the side.
Well, you have the privilege of seeing some of the possible questions that might come up.
Oh, it was Thursday.
What is that? And then see.
Is this still in the same classroom?
And
I don't
you know, all
feel
the
like
fun
it's
stuff actually. Too fun. Okay, so you guys get the idea? 1, 2, 3, 4. And that's how 3, 4, 5. You guys get it.
We get it.
Yeah.
You only saw the extra credit questions.
Yeah, put out extra.
Why would I want to do that?
30 points worth of
We should
extra
put
credit.
your best class in.
Who told you?
Everybody else.
Yeah.
Okay.
Oh, she put something like.
Very good. I think that's
The
got all
greatest
the
American
side
rock.
chains and all that stuff.
It doesn't.
I'll know, see from this, I know, you know where trips and cuts.
Yes.
I know that you know the
that
mechanism, and I know that you know all the amino acids. So technically on the exam, I could probably only put 4 or 5 questions and just pack everything into those. And you'd be able to do that.
No.
Please don't freak out. I'm not eager to do that. I won't do that, you know.
Yeah,
I probably have 7 questions.
That's like punching a dolphin in the
I might do this, but I'm not gonna do like every chapter pushed into
One question.
one question.
That's no money.
You guys can do it. though.
I'm like, bro, I'll die.
Does anybody have any questions? More questions. We got time. We got time. So that's the mechanisms. We've done the kinetics. We looked at the sugars. Yes.
I was just about like those second parts of chapter 7. So about like vaccines and antibodies and then the motor proteins.
Yeah.
That was supposed to be on this last exam.
I didn't
So-
do it, but that doesn't mean I can't.
I know, so.
You should just do whatever's on the test.
It might be there. You've already studied for it, so
Oh, yeah.
it might be. It might
Also,
not
if
be.
it's not gonna be on there, we can just skip over it, that's fine with me.
I know. I know it's fine. A lot of things are fine.
But I mean, you've been emphasizing the study of the last test, and you said it'd be fine. That was on the last test, we should be okay.
I didn't notice this, but there's a television screen on here.
Yeah.
You
can
notice
watch
that-
what you're
Did you
doing.
notice it the last time?
Said that.
I'm a little slow. So it's still free game. Here's what I would say. Okay, so our normal exams are about 6 questions. Here, maybe I'll let you have some info. How about that?
Yes.
We can either do an extra question, like 8 questions, like normal, with an extra question, where it spreads the material out over more problems, or I can cut it down into maybe like 5 questions, where I pack some of it together, kind of like this.
Yeah.
Hey,
Like the A.
hey, let's see Danny.
That's usually what I do. But I thought, you know,
¿Qué preguntó?
if I do it this way,
Do
maybe it'll do that.
5
4 or
questions
5
like this or 8.
Okay, more residents.
Oh, hell no. Hey.
Does it put the points burden on one problem? Okay.
Good solid.
So this is now some of them I have to. Like chapters 2 and 3, like thermodynamics, all that stuff really has to go with the other ones, if you right. But all the other ones, I'll try to keep them sort of distinguished from one another.
Can we vote to veto this?
No,
Yes.
it was not a
No,
democracy.
veto.
Will you go over
I'm a
the,
dictator, and I just gave you some input, okay?
will you go over the extra credit one from the
Do
last
we have
test?
2 extra credits?
What is, oh, so
Oh, the last one was
besides
super
extra
easy.
credits, you have 2 more days to ask me about extra credit. Um, Does anybody have any questions related to the material that I can go over?
What's your favorite one to ask?
I really like this one.
He's gonna include that.
No, I mean, a lot of it, like the enzyme stuff, I'm gonna give you similar questions because there's only so many ways I can write it. So we had mixed inhibition on this one.
Yeah.
Did everybody see that?
Yep.
So that takes a lot longer than say competitive or uncompetitive.
Does that mean you would go through the other ones or do you
I
like
might.
this again?
I might.
No.
Yes.
Wait, Ronald?
Yeah,
If I have a grade, I should have a grade.
as long as you have to know the names. Like Ramachandran.
Nope, you didn't mention it, not even mention it there.
I'm not gonna ask, I'm going to get the second.
Does anybody have anything? I mean,
No call?
I feel like sometimes you come to probe to figure out what's he gonna put on there.
Yes.
Just know it all kind of thing.
That's too much.
That's too much.
We have other findings.
You have other finals?
Yeah.
Heck with those. Talk about life, man.
Could you explain that extra credit one? From the-
Bro, I got 7 pages worth of
I'm sure it was easy.
You attack the red tail?
How do you attack that from behind? If it's-